Everyone can see that? Brilliant. Cool. Okay, so I'm James Bickerstaff, and I'm going to talk about a bit of my PhD research on sympatric speciation, genetic and symbiotic differentiation in the Australian Nestis ambrosia beetles. But before I begin, I'd like to acknowledge the traditional custodians, the Darug people of the Hawkesbury, on which I've conducted my research. I pay my respects to their elders, past and present, and I'd also like to further extend my respects to the traditional custodians on which I've conducted fieldwork throughout Australia, from southeast New South Wales up to far north Queensland. So, ambrosia beetles are a polyphyletic group of weevils. So they consist of the Platypodinae and also up to about 15 independent lineages of scollentines. And so ambrosia beetles are a group of beetles that form a symbiotic association with ambrosia fungus, which they obligately farm as a sole source of food and their resource. For my research, I'll be focusing purely on the Zalsandras clade in the Zilliberine tribe. Oops. So Zilliberines are a group of ambrosia beetles that construct nests in wood or tree branches, and they farm fungus on the tunnel walls. And so if you see in this picture here, you can kind of see like the white fluffy fungus that's growing in the tunnel walls, and that's the ambrosia fungus. And they carry this fungi in mycangia, which you can see in the silhouette over here in this white area. And so it's kind of like an internal invagination and a specialized organ which can carry fungal propagules between nests. And what's really interesting about the Zilliberine ambrosia beetles is that they engage in permanent inbreeding as their primary reproductive strategy. So a gallery or a nest will primarily consist of a foundress mated queen, and also diploid daughters and haploid sons. And these sons are blind, they're flightless, and they're also dwarfed as well. And their sole responsibility in life is essentially mate with their sisters. And once their sisters are mated, they will fly off and found new galleries. And so it's quite interesting that that the males are haploid and the females are diploid, and that's how they really reproduce. And so many Zilliberines have a specialized relationship with their ambrosia fungus. And so in this tanglegram here, you can see quite a lot of associations between beetle genera and also fungal clade. And specifically looking at the Nestis genus, many of these, all studied Nestis beetles so far in farm beaveri clade ambrosiella fungus. And so there's quite a strong co-phylogenetic relationship between beetle and fungus. And so in Australia, we've got quite a good diversity of Zilliberine ambrosia beetles. However, one of the most widely distributed Zilliberine ambrosia beetles is Nestis. And so we've got, in Australia, we have two species of Nestis, Nestis solitis and Nestis pseudosolitis. They have a sympatric distribution from Tasmania to far north Queensland. The host tree generalists, so they're typically found in eucalypt trees, but also they've been found in quite a lot of other native tree species, including other crop plantation species such as macadamia, apples, or cherries. They integrate in morphology. There's very little morphological differences between these two species. And Shadle in 1936, when he was describing Nestis pseudosolitis, primarily identified this species by the darker color and also the abrupt elytral declivity. However, later taxonomic research suggested that there's essentially no morphological differentiation between the two species, and the species names are presumed to be synonymous. And also, we don't really know what species or clade of Ambrosiella nestis are farming in Australia. So, as part of this research, we investigated what species of Ambrosiella fungi Australian nestis species are farming, and also we tested whether or not the Nestis solitis and Nestis pseudosolitis names are synonymous with one another. So we collected ambrosia beetles throughout Eastern Australia, from southeast New South Wales to far north Queensland, and we also accessed specimens from Tasmania from the Tasmanian Museum. We sampled ambrosiella fungus from the mycangia using ITS sequencing throughout the range using specialized primers, and we also cultured fungus collected from Cedar Ridge. We also used beetle population markers, we used cytochrome oxidase 1 and arginine kinase to assess the population structure of Nestis throughout Australia. So primarily, we found two Ambrosiella symbionts associated with Nestis. Throughout the entire range from northern Tasmania to far north Queensland, we primarily found Ambrosiella species 1. However, in one population in southeast New South Wales, southeast forests, they were farming a highly divergent species of Ambrosiella, which we named Ambrosiella species 2. And we managed to culture Ambrosiella species 1 from Cedar Ridge as well. Interestingly, however, these two symbionts are quite differentiated from one another, and they're also found in different Ambrosiella clades as well. And so going back a few slides, previous researchers identified that Nestis is primarily associated with beaver eye clade Ambrosiella fungus. However, in our Nestis solidus, we found that Nestis solidus is primarily farming Ambrosiella species 1, which is found in the Zillabori clade, which is quite unique for the genus. Interestingly, in the one population in southeast forests, this species was farming Ambrosiella species 2 from the beaver eye clade fungus, which kind of suggests that there's a Ambrosiella symbiont switching going on, or potentially a selective sweep for Ambrosiella species 1 throughout its range. And so when we started investigating the population structure of Nestis solidus throughout Australia, we encountered Wolbachia using LCO-HCO primer sequencing for cytochrome oxidase 1. And as mentioned previously, Wolbachia is an intracellular symbiont. It's found in a wide range of insect species, up to about 60% of all insect species. And this endosymbiont is quite interesting because it's also a reproductive manipulator, which can induce cytoplasmic incompatibility, wherein if one individual is infected with Wolbachia and it mates with an individual not infected with Wolbachia, the cross will not work out. And it can also induce Thelytochus parthenogenesis, wherein the infected population or species will be able to produce diploid offspring. And as Wolbachia always wants to reproduce throughout populations throughout time, it will promote the production of female offspring to carry Wolbachia throughout populations. And this can cause this can cause widespread mitochondrial selective sweeps as well, really greatly affecting the mitochondrial diversity in their host populations. And so to assess Wolbachia presence and identity in Nestis, we screened individuals with the Wolbachia surface protein primer set, and we also identified the strain using MLST approaches. And so with both WSP and MLST approaches, we identified a novel strain of Wolbachia associated with Nestis solidus, which we termed WC solid. And interestingly, this strain of Wolbachia was quite closely related to the strain infecting Drosophilus simulans from Hawaii. So, I went the wrong way. And so potentially there might have been a bit of a holiday that Nestis took, but who knows how really it got here. And so looking at the population genetics of Nestis throughout Australia, we identified I'm getting my words mixed up here. We found 2 genetic lineages of Nestis throughout Australia, and we identified both Nestis pseudosolitis and Nestis solidus within each lineage. And so we can confirm the synonymy of solidus and pseudosolitis. Interestingly, there was little nuclear divergence between the lineages, and there was quite a lot of mitochondrial diversity between the lineages. However, there was no nuclear or mitochondrial diversity within lineages. We also found Wolbachia only associated with lineage 1 Nestis solidus individuals, and this lineage 1 also had a high prevalence of captured beetles as well. And then we also found no Wolbachia associated with lineage 2 Nestis solidus. And so these 2 cryptic lineages are distributed in sympatry throughout Australia. However, there's quite a high predominance of lineage 1 infected Wolbachia lineages, beetles, within Eastern Australia as well. And so this all comes to a really interesting test or hypothesis about how these 2 species, or how these 2 lineages are cryptically speciating from one another in sympatry. And so from this research, we can confirm the synonymy of Australian Nestis species. And we've also found that both lineages are distributed in sympatry as well. However, we don't think that ecological mechanisms have generated the diversification between these 2 lineages, as we identified both lineages are host generalists with their host tree preferences, and they also found the same strain of Ambrosiella as well. And so we don't expect niche divergence has led to sympatric speciation in these 2 lineages. We predict that permanent inbreeding will reinforce the sympatric distribution of these two species, as they both engage in permanent inbreeding as their only essentially reproductive mechanism. And so we don't expect that individuals from lineage 1 or lineage 2 will cross with one another. We also identified that Wolbachia, we also predict that Wolbachia may play a role in the sympatric diversification of these two lineages. We identified these two genetic lineages with highly different mitotypes, and there was also no mitochondrial diversity within either of these lineages either. And so, given that we found no mitochondrial diversity within either lineage, we don't expect that Wolbachia has caused reproductive manipulation in either lineage through either cytoplasmic incompatibility or parthenogenesis, as you'd always expect a higher mitochondrial diversity within the uninfected lineage. And so we predict, or we want to test, that potentially there's a potential fitness benefit conferred by Wolbachia in the infected and highly prevalent lineage. And so I'd like to thank my advisors, Marcus Riegler from Western Sydney University, Hawkesbury Institute for the Environment, and Beate Jordell from the University of Bergen. I'd also like to thank Western Sydney University and the Holsworth Wildlife Research Endowment for funding this research. And I'd also like to thank TMAG, CSIRO, ANIC, and QDAF for providing samples for my research. Thank you.
Thanks, James. We've already got a question from Perry. So are the beetles plug-shaped for a reason? Do they block galleries with their bodies?
Yes. So in many species of ambrosia beetles, it's been found that they can use their elytra and their butt essentially to plug gallery and tunnel holes to protect the gallery or the nest, and to also prevent interlopers coming in. And so what you see in quite a lot of ambrosia beetle species, they've got these highly modified elytra to protect the main entrance gallery.
Excellent. Ari Hoffman has a question.
Yep.
Yeah, hi James. Look, great talk. Really interesting system. Thanks very much. I'm trying to just think about this mitochondrial sweep, of course, and be involved in the Wolbachia. And so you're suggesting a little variation in a species or a lineage, suggesting a Wolbachia sweep of some sort with a hitchhiking potentially, but you don't like that idea, if I understand you correctly. But of course, a fitness benefit will still lead to a Wolbachia sweep, right? And of course, we've shown that beautifully in Drosophila, where in one species we have pretty much had a Wolbachia increase that was quite sharp, simply due to a fitness benefit, right? So I think a fitness benefit can produce a Wolbachia sweep as well, which would then drag the mitochondria along with it.
Yeah, we've been thinking about the same thing too, that there's been either selection for the Wolbachia or selection on the mitochondria, which has been driving the prevalence of Wolbachia in the infected lineage. However, what we don't see is the diversity in mitochondria in the uninfected lineage either. And so we don't really know what's causing asymmetry in either lineage.
Right. But you could, of course, have a Wolbachia sweep, and then the Wolbachia is lost for some reason, right? I mean, we have seen the loss of Wolbachia in a couple of situations, well, after a sweep has gone through. So
potentially,
I mean, I'm not saying you're wrong. I'm just saying I just want to keep the hypotheses a little bit open, I think.
Oh yes, definitely, yeah. Because this was very much a study, like we just wanted to investigate the population structure of this interesting haploid diploid ambrosia beetle. Then we found Wolbachia, and it's just, this is a little happening here. What's going on? So it's definitely open for further research. I agree.
Yeah, very interesting. Yeah, thank you.
All right, so we probably have time for one more question. And I'll get my slides going. I guess just as a note, James, actually, the bees I'm about to talk about actually have the same WHA Wolbachia.
Really?
Yeah.
It's getting everywhere.
It is. It's prolific. Alrighty.